All right, everyone. So, good afternoon and welcome back. We are now ready for announcing the winners of today's first day quiz, right? So, day one quiz session, toppers list is shown here. And I'm going to read actually each one of your name. You are putting so much effort, at least I should know you by name. In case you are available right now online and you can turn on your video, we can see who you are. That will be helpful for our future interactions. So I'm just going to read the names for the junior quiz toppers here. Uh, the first winner is Parvesh. Next is Rohit Katemani, and I think you can see the scores are very close. Kashvi, Sonachitra, all three of them are getting same marks. Srinidhi, Maitrey, Kanak Sangvi, Ishan Ghosh, Janvi Chandra, Varsha Rani, Pratyush, Indu Singh, S. Nitya, Sanvi Pandey, V.M. Sakshi, Tanisha S., Chirag Pathania, Sukhmanpreet Kaur, Gabriel Stephen, Sankhanil Chatterjee, Parth Chabra, Krishna Rakshit Nevalia. All right, congratulations to all these junior quiz winners. So you played very well. The questions were from different, previous sessions and general understanding. So it was not something which you can prepare so much, but from your overall knowledge and understanding. So well done. And large number of students have really played for these quizzes. Here the cutoff kept was 65% for selecting the winners. Now let's move on to the middle group. So in middle group, we have Niranjani Ramakrishnan, perfect score 15. Dvija M. Dave, second winner. Ashish, Minal, Shravya, Nirmal, Kashish, Somangi, Aditya Gupta, Neha Kumari, Aradhya Singh, Rutuja, Shravani, Deepika. All of you are today's winner in the middle group. We had several people who have actually still missed out the right categories, and those have been unfortunately disqualified. If the seniors have played in middle group or some other people have played in a junior group, so they were not considered. So in the middle group, we had actually gone 50% cutoff given the, many people got disqualified. Senior quiz, we had a really huge number of participants who played for it. And we had to be really, make a very stringent cutoff here of 80%. But let me still, go through the names. So Anushka, Ishita, Anuha, Narmada, Karen, Ankur, Semanti, Senolina, Udita, Rishav, Shlok, Sucheta, Shubhiksha, Ritbhash, Suchitra, Sai Rohit, some name Freshma, okay, Freshma M, Hardik, Ushma, Akarsh, Megha, Sulagana, Upasna, Andrila. All right, congratulations to all the winners of today's quiz from junior, middle, and senior group. Well done. And of course, you'll get certificates and some award prizes for these performance. Now, let me start sharing you the videos of the different categories. So we had announced for 3 different type of competitions. And for each of the competitions, we had asked for the entries in junior group, middle group, and senior group. The first competition which we are going to play today was essentially to showcase your projects. If you have done some science project, any specific topic in the past, and you'd like to showcase here. So that's something which we are now going to show, and starting from the junior and the middle groups. And then we had large entries from the senior groups. I will show those videos as well. Yeah. So we are starting now the videos of different students who have played from the junior group and the middle group.
I'm Swadami, and today I'm going to perform two amazing experiments using salt and water. The apparatus includes a metal zipper, a 9-volt battery, a glass full of water, salt, some earbuds, crocodile clips, a stencil, and a duct tape. So now we are going to pour some water in an empty glass and make a saturated brine solution. Dip some earbuds in our brine. So now we'll attach the red clip with the positive electrode and its wire with the zipper. Attach the black clip with the negative electrode and twirl its wire around the earbud dipped in the brine water. Here I'm sticking the duct tape on the zipper, one below the other. Now I've cut out the required shape, and now using the earbud, we are going to rub it for 5 to 10 minutes. As you'll notice, the metal is corroded due to the brine and the color is faded, making a beautiful design.
Newton's grid is a device that demonstrates the law of conservation of momentum as well as the law of conservation of energy. It was basically a scientific toy which was invented by Marius J. Morin. So when you lift one ball and release it, it strikes the other balls and transmits the force to the balls, which lifts the last ball. But there's a lot more going on here than just conservation of momentum. If a ball strikes with velocity v, then why don't two balls come off the end with a velocity v upon 2? I mean, the momentum would still be conserved in that case. The reason for this is that when these balls collide, they compress a little. If we take these collisions as infinitely fast, then the first collides with the second with the third, and so on till the last ball. Now, the last ball has nothing to collide with, and hence it is raised. So what will happen if we release two balls? Let's see.
From Gateway the Complete School, grade 10, and my science project is DNA extraction from spinach. First, I blended 25 grams of fresh spinach, which helps in mechanical agitation and cell wall lysis. Then I added 10 ml of dishwash soap, which helps in cell membrane lysis due to the detergent. Then I added 1 gram of meat tenderizer, which contains protease, which breaks down protein and protects the DNA upon release. Then I added equal volume of rubbing alcohol, which helps in the DNA precipitating. The DNA precipitated is white in color and complex structure. From this I learned the steps of DNA extraction.
Idea which is named laser-based danger alerting security system. It is based on a laser. And why do we need it? We need it because we all know that with the advancement of technology, criminals have also come up with new ideas to fulfill their deeds. So, to protect our homes, industries, business places, factories, or even us from thefts and their criminal acts, we need a security system which is advanced and modern. So, laser-based danger alerting security system is just the right choice. What will happen if someone enters a place where this security system is installed? Then he will disturb the laser because the laser is almost invisible and it is visible only at the point of source and the incident point. Now, if it disturbs the laser or stops it,
according to WHO, every year there are more than 1 million deaths caused due to mosquito bite. So what are the measures to prevent mosquitoes? Products available in the market like coil, roll-on, repellent have health hazards. I'm going to present my project on intelligent solar mosquito control. We did this group project to make a clean and cost-effective solar energy-based solution for mosquito control. This is our model. Suppose this is a room and this is a window. The window will have a mesh behind it. It will have 3 layers, so the inner layer will be heated, and when the mosquitoes try to enter our house, the middle layer will automatically allow the current to flow and the mosquitoes will die when they enter through the mesh. The variety of the work is that it can work for 24/7 and even at night. It kills the mosquitoes quite fast using human safe low voltage electric.
tablets for food grain storage in-house. Myself, Nirmal Chopre from Jawahar Navodaya Vidyalaya Sangli, studying in 12th class. When it comes to storage of food grains in rural areas, it is a difficult task by the use of modern technology and chemical pesticides because either they are very costly, unhygienic, and not easily available in rural places. Due to this, there is tremendous loss in food grain, lead to malnutrition, and unhygienic to meet the demands of growing population. By using waste-generated material by these products, which are easily available in my house. So I put these bio tablets in these containers and later I found out these results. The bag containing tablet A is free from any pest and the bag B and C containing tablets show a little number of pests. Here are the applications of projects.
Quasar. Quasar is a luminous AGN which consists of a supermassive black hole surrounded by a gaseous accretion disk. It is among the brightest and most distant celestial objects. A quasar is approximately 1 kiloparsec in width. Its properties depend on many factors. Other classes of AGN include Seyfert galaxies and lasers. When compared to a black hole, quasars have clouds of gas around the inner structure. They are known as young and active galaxies, which are extremely bright and are very far away from us. The oldest quasar is at a distance of 13.03 billion light years, and thus we see it as it was after 670 million years after the Big Bang.
All right, so very exciting, a lot of interesting ideas and good projects done by the junior and middle group students. In case you were the one who had submitted a project before the deadline and we are not able to show your video, please do email me and send me the link where you have uploaded the video so that we make sure that you are part of the contest, right? But these are all the videos which we have shown. Now, of course, some of the winners will emerge out from these competitions. Let me now go to the senior group, which is much difficult one to go through. A lot of videos are there for that.
So the topic of my project was the Big Five personality traits, effect on stress response and preparedness during the COVID-19 pandemic. So these are the 5 personality traits that come under the Big Five personality traits, that is neuroticism, extraversion, agreeableness, conscientiousness, and openness to experience. These were my hypotheses. So I used a survey, I developed the questionnaires and I used a survey on Google Forms to collect the data, and then later I performed the statistical analysis via Spearman's rank correlation test. So the results that I obtained were that highly neurotic individuals experienced more stress and were more prepared. Highly extroverted individuals experienced less stress and were less likely to comply with social distancing norms, whereas agreeable individuals experienced less stress and were more likely to engage in altruistic behaviors. Conscientious individuals experienced less stress and were more prepared for the pandemic. So this could help us to take a more targeted approach towards stress management during next pandemics, and also a targeted approach in the communication of the mitigation strategies to the public. That is, mitigation strategies such as hand washing and wearing a mask. Thank you.
Pharmacy Biotechnology in University of Madras. I'm going to present my final year project. The title of my project is Comprehensive Gene Screening Using Clinical Exome Analysis Resolves Genetic Etiology of Hearing Loss in a South Indian Family. Samples of the families that are screened negative for common deafness-causing genes are taken from the consortium. Here, our aim is to screen the genes that are responsible for causing deafness in hearing-impaired family. The variants are to be identified using clinical exome sequencing, and the same are confirmed using Sanger sequencing after PCR amplification. These are the methods that are involved in my project. The clinical exome sequencing results showed variants of DIAPH3 gene. The location of the variant is predicted and the primer is designed for that particular exonic region. The samples are then subjected to PCR and with the designed primer. The amplification of the particular region is confirmed using agarose gel electrophoresis, as you can see from the gel image. If confirmed, the samples are given to Sanger sequencing, and the results of the Sanger sequencing are analyzed using Finch TV. Further bioinformatics analysis is also done.
Hello, my name is Gunja and I am doing my BSc Microbiology final year from JD Goenka University. I did a small project on the topic of evaluation of microbial parameter of drinking water near Delhi region. In this project, we collected a sample from 4 different areas: Gurugram, Sohna, Palwal and Manesar. We have to check the microbial density and population of that water that people is using for their household and many purpose. Then we collected a sample and we grow that on a mechanical and nutrient agar medium, and we observe that which area sample water is highly contaminated with the microbes. And then we come to the conclusion that industry area are highly contaminated with the microbial population, and we presented our idea to the international conference, and for this we also got the best idea presentation award. Thank you.
Extraction identification and silico analysis of bioactive compounds of Rhinacanthus nasutus against Alzheimer's, diabetes, and inflammation. The botanical name is Rhinacanthus nasutus. It belongs to the family Acanthaceae. It has many medicinal properties. From the extraction process, I got aerial and root extract using ethyl acetate solvent. In order to check whether my sample is phytochemical compounds, I did the phytochemical assay. Same goes for the antibacterial activity. The stars indicates the activity in different concentration of the extract. Biochemical activity assay for Alzheimer's disease. The antioxidant assay shows to have higher scavenging activity in aerial extract, and for neuroinflammation, I did the nitrate assay where the root extract shows to have higher activity. Docking was done by beta secretaries and Alzheimer's marker and Rhinacanthus compound, which is present both in root and aerial. Glucose 6-phosphate dehydrogenase, a diabetic enzyme marker, docked to Rhinacanthus. This is the interaction scores for each enzyme. The binding energy is higher for beta secretaries, followed by glucose 6-phosphate. For the works are going on in this beautiful plant.
Hello, biology summer camp team from IIT Bombay. I'm Jitul Dave, a graduate zoologist and doing a master's degree in zoology. I have done research on the foraging behavior of a jungle babbler. It is a residential bird in my city, Jamnagar in Gujarat. And I have just studied on the best preferred food consumed by the jungle babbler in its breeding and non-breeding season. And I have included 4 different type of grains, and from which a particular grain is mostly preferred in its breeding and non-breeding season. And in non-breeding season, I got the peanut, and in breeding season, I got peanut, but they actually feed on insects rather than the peanuts, because it is breeding season and they need some more energy other than the peanut. And other than that, the research is just gone on for the 8 months, and that's all about my research. Thank you very much for this opportunity to share my research.
Hello, I am Maitreyi Balme, second year BTech student of IIT Guwahati from the Department of PSB. Currently, I am working under my professor Dr. Sachin Kumar in his viral immunology laboratory. And recently, we studied the effect of cholesterol on anatid herpes virus 1 infections in vitro. So yeah, the effect of cholesterol on the host cell membrane and the viral envelope both on the infectivity of ANH1 was explored. We performed a series of experiments, and the result implied that the cell membrane cholesterol is vital for the infection of ANH1, and its depletion from the virion curtails the infectivity of ANH1 by destabilizing the envelope. And yeah, the paper even got published by Elsevier in 2020. Thank you.
I am Nathan doing genetic engineering at Bar Institute of Higher Education Research. I would like to tell you about the project that I performed earlier. So main theme of the project to treat carpet-axle at the industry. As the project done few years ago, unfortunately I could not show the project. So the project actually consists of a small home-like apparatus, which actually industry, and we have the kerosene inside, which burns and emits the black gases into a tunnel. So a motor which circulates the water, which also a looping. So we fixed the looping water pipe into the tunnel, so that the air which consists of toxic CO2 and CO fuses into the water, and the water gets looped. So the gas outside the tunnel will be less toxic compared to the original one. Thank you.
This presentation is based on methods to rejuvenate Northwest Rajasthan using nanoclay and halophytes. The aim is to reduce the salinity and improve the water and nutrient retention capacity as well as the nitrogen and phosphorus content of the soil by using ammonium phosphate-rich nanoclay mixture. We can reduce the salinity by using calla grass, which is known to accumulate salt from the soil. The process for making nanoclay is time-consuming but rather simple if the methods, as mentioned in the slides, are followed. After getting the nanoclay, we can mix in ammonium phosphate and water and can apply the nutrient-rich liquid nanoclay directly on the fields.
There are a few more videos. I'm just opening the last one.
Good morning, everyone. I'm going to present my undergraduate research work. We all know that the brinjal has both medicinal and nutritional values, but nowadays it is very prone to damage by the insect Leucina rhodiominalis. Thus, farmers are using various kinds of chemical pesticides in the agri fields, and you may know that using these chemicals is very harmful for both the human being and for the environment. Thus, we tried to evaluate the entomopathogenic efficacy of the isolated fungi, and for this I performed the soil collection, identification of the genus, in vitro screening, TLC, enzymatic assay, and as a result of this research, I found that the both Penicillium, Aspergillus, and the Fusarium has the highest capacity to get the protein, and my TLC result suggests that the Aspergillus chemicals has a structural similarity with the Vivericin, that is a potent insecticide. So, in conclusion, we can say that using this fungi against this as a biological control could be a very meaningful approach, but more research is required to establish this statement. Thank you.
Good evening. I'm Pinaki Chattopadhyay, Department of Botany, West Bengal State University. I am here to deliver a short speech on protein extract crystallography. I have done a project on protein crystallography. Actually, it was a skill development program from CSIR IICB, Kolkata. And here, where we learned about how to determine the three-dimensional structure of protein with the help of passing X-rays through a protein crystal, and after that the results we got the dots. On the dispersed dots we can diffracted the dots, we can do Fourier transform, and with the help of some bioinformatics tools we can demonstrate the protein structure, and also we could find the active domain of the protein, means which is the actively participating portion of the protein, and it is very useful, and it is a very means huge range of application. It has various range of application in enzymology. Thank you very much.
Hello to everyone. My name is Pooja Reddy. We the normal people, we have ample number of opportunity in field of education. But what for the blind people? They are lacking in the field of education. Because of the braille display which was present in the market, they are highly expensive due to the manufacturing of actuator which is required via arithmetic. The technology which is used in a current actuator is piezoelectric, magnetic, and shape memory alloy. So we are introducing new technology which more effective and reduce the cost of actuators. So we are researching on the two technology. First is our mechanical actuator and second is our dielectric elastomer actuator. This is our mechanical actuator. Talking about achievement, we are the winner of Sardar Patel Technology Business Incubator and a government competition called Toykathon.
Everyone, today I'm here to talk about DNA extraction, a routine procedure in biotechnology. DNA, also known as deoxyribonucleic acid, is a double helix structure of two polynucleotide chains which contain all the genetic instructions required for development, function, growth, and reproduction of an organism. Isolation of DNA is required for a variety of applications in science, medicine, and forensics. For example, scientists introduce DNA into the cells of animals or plants for SNP analysis, DNA methylation analysis, Southern blotting, PCR, etc. In medicine, DNA isolation can be used for diagnostic purposes. Also, in forensics, DNA isolation is used for the identification of individuals such as rapists, thieves, etc. Also for paternity determination and body identification. The procedure for DNA extraction, while different, follows three basic steps. They are lysis, precipitation, and purification. I have done this project personally as a part of my 12th standard biology practicals and extracted the DNA from strawberries. Thank you.
So my name is Sally Samant. I'll be talking about my third year research project on functional analysis of the EGFR network using gene ontology. So the basis of this project is a systems biology paper on the EGFR network published in 2005. We tried to identify functionally important terms, the gene enrichment terms, gene enriched terms from the map using gene ontology, which segregated each biological term on these three bases. We used David, which was a software of annotation, visualization, and integrated discovery for doing this. And we performed a functional annotation analysis through which we generated 13 clusters. You can see 14 here, but one was non-significant. This was done at the highest stringency, and we developed a legend to show the 14 clusters. This helped us in identifying gene-enriched targets, which can be used as therapeutic agents. So this is all from my end, and thank you so much for the opportunity.
Hello everyone. In this video, I am explaining about my BSc project and my
Here I am Shomodipto Shaha from Maulana Azad College, Kolkata. Here is my presentation. Here is a mathematical model based on non-Euclidean geometry. Such a geometrical study of arbitrary shaped things. We all know that before Pangea happened, all of the world continents may attach to each other. Here, we took America, Africa, and South America. At first, we took various lengths of lines, such as 5, 4, or etc. centimeter line. Then by pencil compass, we take those of the length one by one and cut piece of coastal line of both of the continent. These are the value provided here. By putting this value on the graph, we get almost to same curve. So it's proof that the two continents may attach to each other before Pangea happened. Thank you.
Good morning, everyone. My name is Tamayal. I'm an undergraduate bioinformatics student. Thank you for giving me this opportunity. I have done this project on my final year project under the guidance of Dr. Subha Mohan. So my project is based on the transcriptomic guided drug repurposing for COVID-19 treatment, a meta-analysis based approach. So these are the methodology. We collected the publicly available data sets. And there is an online tool for meta-analysis for network analysis tool. We taken the RNA-seq and microarray data for integration. We use the combined effects axis. Based on we compare with the differentially expressed genes for the other respiratory infections. So based on which we kind the proviral factors. And for drug repurposing, we use the online tool for flu. So there's an online tool used for the drug preparation. So these are the results we obtained from this. This is the protein level interaction analysis, CMAP analysis for HLX. There are 12 of them are proviral factors that reverse the activity. Thank you.
Greetings, my name is Ushma Pandya. For my undergraduate project thesis, I worked on characterizing and modifying 4 enzyme sequences using available computational tools. These enzymes were novel microbial histidine acid phosphatases. The objectives of this study were firstly to carry out in silico characterization of the 4 sequences and do a comparative analysis. This was achieved by carrying out a primary, secondary, tertiary, and functional analysis of each of the proteins, along with an evaluation of the simulated 3D model. The second objective was to carry out rational design protein engineering using the mentioned computational tools by introducing single and multi-point mutations into the protein sequences to increase their structural and thermal stability. My teammates and I were successful in creating mutants of each of the proteins that showcase greater stability than their wild-type models. Thank you.
The topic for today is antibiotic sensitivity testing. Whenever a bacterium is found to be responsible for causing infection in the human body, antibiotics are administered to fight them and finally cure the infection. Before an antibiotic is given to a patient, it's imperative to know whether that antibiotic is actually effective against that particular bacterium. The method in the lab used to verify this is called the Kirby-Bayer method of antibiotic sensitivity testing. A suspension of the bacteria is made whose turbidity equal to 0.5 McFarland standard is spread evenly over the Muller-Hinton agar plate. The disk of antibiotics to be tested are placed and the culture plate incubated at 37 degrees Celsius for 24 hours. If the bacterium is sensitive, a zone of inhibition is formed around the disk. The zone size is measured and compared to the standard charts, and the result is obtained as sensitive, intermediate, or resistant. The results to my experiment are as follows.
Hello, my name is Yash. I'm a student of first year B.Tech Biotech from D.Y. Patil School of Biotechnology and Bioinformatics. Today I am sharing the project which was created by me and my friends in the year 2017. Our project was a cleaner bot. The main reason behind developing this bot was to automate the process of cleaning. The bot was programmed on an Arduino chip with basic sets of functions. Sensors were installed in front and back to identify the obstruction present in its way. The whole system was powered by a 10,000 mAh power bank. The major part was the vacuum system present at the rear. The vacuum unit was powered by a separate power source which was a 4,000 mAh power bank and had a 3 to 9V high-speed DC motor for vacuum purposes.
Quasar Kudineer, KK, is a traditional Siddha formulation and has been prescribed in Siddha medicine for the management and treatment of flu-like symptoms. In the study, we focused on using computational methods to identify host targets in different pathways where the phytocompounds of KK possibly interact. Specifically, we used a network pharmacology model coupled with molecular docking to analyze host targets. Using the curated list of phytocompounds from 15 plants obtained from existing literature, each compound was searched against the PubChem database to derive its SMILES format, using which the probable host targets were predicted in Swiss target prediction and binding DB. Probability scores of 0.5 and above were classified as high confidence targets and the others as low confidence targets. We constructed a network of compounds and their possible interacting targets and identified 3 pathways that the compounds possibly modulate. Then we performed molecular docking simulations for the phytocompounds to map the molecular interactions and ascertain their role in antiviral treatment. And we found the best binder to the targets of these 3 pathways.